My function title is program manager, and that means that I am content aligned within our organization, and my domain is proteins and what we call the protein transition. So I'll just walk you through some of the things that we're doing, some of the things that I think are interesting new developments here, and hopefully leave some time for questions at the end. But before I do that, I did want to spend just a few seconds explaining our organization. So as you see here in the bottom part of my slide, we refer to ourselves as Vāgeningenas University and Research. Helpful to know is that we do have a university, so we are an academic institution, but in parallel to our academic institution, we also have contract research in all of the science domains that are also represented in our university. And we are a rather unique institution in that we are fully focused on life sciences, as you see in our overall mission, which is to explore the potential of nature to improve the quality of life. We were founded as a food and agriculture institution 102 years ago, and that's still at the heart of what we do. In my personal domain of proteins, our vision is working toward a world with sufficient, nutritious, delicious, and sustainable proteins for the growing global population. I think that's something that many companies and many institutions are working on, and a sort of shared goal that represents, as you said, a hot topic at the moment. We focus on 5 areas where we think as a science-based institution we can help. Number 1, we work to accelerate the development of new protein sources. And I was asked to talk a little bit about some of the ones that we are working on, so I'll dive into that in a moment. We're also concerned about protein extraction efficiency, you know, the energy that we put in to get the proteins out of some of the sources that we're using today is, you know, detrimental to the environmental footprint, takes a lot of energy, a lot of water. We think that that can be better. I won't focus on that very much today because I understood the interest here is more on new sources, and then on plant-based foods. So that's the next one, improving the quality of plant-based foods, you know, the meat analogs, the dairy analogs, but also looking a little bit broader nowadays. I'll focus on meat and dairy since those are the 2 biggest categories. Number 4, we also work on providing nutritional quality data. So we do a lot of characterization of proteins for their bioavailability, for example. I don't focus on that today, but just that you know, that's something we are working on within our organization. And then last but not least, we support companies who are taking a new protein source through the Food Safety Authority approval process. So I'm going to dive in a little bit to number 1 and number 3 today. If we start by looking at new sources, I just picked out 3. This is in no way exhaustive. I mean, there are as many protein sources as you can count these days, and all sort of being investigated in different places and for different reasons. But these are 3 that represent for me also 3 interesting sort of buckets of proteins. You know, what do we have that's relatively short-term accessible to us here in Europe? What do we have in the medium term that's sort of coming through the pipeline, but very interesting? And what do we have sort of long term? What do we expect to see? So the first category are pulses. I don't know if you recognize that picture. Those are fava beans. Well, and we see that pulses, you know, there's a lot of energy around this topic within Europe, and for good reason. These are interesting crops that can be locally grown efficiently in different parts of Europe. They provide, you know, good functionality in plant-based foods. They bind nitrogen into the soil, so in that way they also reduce our dependence on fertilizers. They do have some challenges in application today, so I'll talk a little bit through what those challenges are and what the potential is and how we're working on those. Second category of proteins that I personally find really interesting are from agricultural and industrial side streams. These proteins are typically categorically different than the plant storage proteins that we are using from the pulses, which means they can also fulfill some really interesting functional niches. And I'll dive into crop residuals, which is a specific subcategory of those side stream proteins. And then last but not least, a category that's maybe a little bit longer out, but nonetheless what that we consider really interesting are fermentative biomass proteins, by which I mean we're growing a bacteria or a yeast, and we are, you know, we're not using that as a kind of cell factory. That's a whole other technology, but when we're using the bacteria or yeast that we've grown itself as a source of protein. And I'll talk you through some of the things that are in development in that regard. So let's start with the pulses. One of the big challenges that we see with pulses, there are a few, but one of the big challenges that we see is to do with flavor. Again, if you look at those fava beans, they tend to taste like a fava bean, which is wonderful if you're sitting down to a plate of fava beans, but if you're sitting down to a plant-based yogurt, that's not what you expect from it. Same with peas. And most of the pulses, they have a pretty characteristic taste, and that sort of taste and flavor composition typically comes along with the protein when we do an extraction. Of course, the composition does vary depending on the varietal, depending on the growing location, on the crop year, but we can speak in general terms about off flavors like green, beany, pea, earthy, or hay-like, and then taste aspects, taste defects, typically sort of bitter or astringent. And by and large, we know what compounds are associated to causing those kind of off flavors, the aldehydes, the pyrazines, and the saponins for the bitter astringent perception. The fact that we do know which components are causing this actually presents us an opportunity. These pulse crops have been cultivated already in Europe for a long time, but we've never optimized them for the purpose that we're using them for now. If you take peas as an example, the peas have been cultivated for their starch. Pea starch is a typical industrial food ingredient, but we've never optimized them for the protein, and we've certainly not optimized them for a taste performance, a clean taste in, for example, a plant-based yogurt. So we're working on different fronts to try and address those sort of issues that we see in flavor in the different pulses. In the first case, some of those off notes that we see come from oxidation, which is during processing, and that's a relatively easy one to tackle. If you have an oxidation-related off flavor, like beany and soy can be related to oxidation, you can optimize the process of extracting the protein to eliminate that off flavor. If that's not the case, and we see that, that the off flavor is really coming from the plant itself and not from the process, there's not a lot we can do to optimize the process, but we can do fermentation to remove specific off flavors. We can even use fermentation to convert a flavor into a positive flavor and off flavor into a positive flavor. And we do that by matching a specific microorganism who can consume that flavor using a high throughput screening to match the substrate and the desired outcome with the organism. Last but not least, but really important, and one that I think is long-term the most promising, even though it takes us some time, we have a collaboration between our food technology group and our plant breeding group, and we are using that collaboration to breed pulses which have fewer off flavors inherent in the plant. And you see just the little circular diagram of how that project works. We're identifying what the requirements are in the end product. We're screening our cultivars. We're sort of breeding the most promising pulses. Then we're testing that in real life through the process, because you can imagine if you optimize for a clean tasting fava bean at the top of the pipe, that doesn't mean you're not creating off flavors by the end of the pipe. So this sort of circular process whereby we're breeding and then we're checking each time through a real model food process. If we jump to the sort of midterm protein source that we think is very interesting, I'm going to talk a little bit through crop residuals, by which I mean agricultural side streams and sort of winning proteins back from those. We think this is very interesting because it increases our European supply without requiring new land. It helps farmers by strengthening their their business case, basically. And as I said, these are categorically different proteins, so they provide sort of unique functional and in some cases nutritional benefits. And in particular, I'm going to tell you a little bit about rubisco, which is the world's most ubiquitous protein and is present in all green leaves. This picture of a field that you see, these are sugar beets, which we cultivate for their sugar, kind of obvious. And when we cultivate that sugar beet underground, we also get sugar beet leaf above the ground. And that's an example of a material that has is an interesting source of rubisco protein. Another example, potato proteins, for example, are a very interesting category already on the market, having been industrialized in the first case, industrialization of potatoes were happening for their starch. But nowadays that business case is completely flipped on its head, and potato proteins are earning all of the all of the money for the potato cultivating companies. So if we do dive in and look at Rubisco, as I said, I'm not going to go in great detail on the functionality of this, but I'll just show you these two graphs of Rubisco gelation from sugar beet leaves and from spinach, and comparing that to some other proteins that are typically used for gelation, whey protein, which comes from dairy, of course, and soy proteins, and then the spinach one also compared to egg white. We think this is very compelling to say these are really interesting functional proteins, because you can get a gel at even lower concentrations than you see with whey or egg white proteins at neutral pH, and they have the highest gelling properties of the many different plant proteins that we've compared them to. And that's also consistent across multiple raw material sources. If you compare the sugar beet leaves and the spinach, you see a pretty consistent behavior there. Typically, at the moment, the Rubisco proteins are not available on the market. Just to give you an update on the status, the sugar beet leaf protein has been submitted to the European Food Safety Authority, and we expect to see that coming on the market in the next couple of years. Then if we look a little bit longer term, as I said, I think fermentative biomass is something we consider really an interesting low-cost option for the long-term future. And by that, when I talk about fermentative biomass, some people refer to it as single-cell proteins. I mean, for fungal, bacterial, or yeast-based sources. You can grow these on all kinds of different substrates. We can grow them on agricultural residues. We can grow them on pure glucose. There are many innovative startups who are even looking further beyond that, the so-called sort of air proteins, which are grown usually using a hydrolysis process. Also very interesting that these sources can be integrated into no-waste energy food production systems. So we're part of a European project where we're creating biofuel and using the actual residual material from biofuel to grow protein as biomass, biomass as protein. We also believe that there's high potential here because the biomass can be much closer to meat, both in terms of texture and in terms of texture, the mycoproteins are a good example there, but also compositionally. So most bacterial biomass can have 40 to 60% protein, which is much closer to meat if you compare it to a plant like a chickpea, which has about 17% protein in it. So mycoproteins, that picture is a picture of some mycoproteins. They're, as I said, the ones that are sort of most developed. If you've eaten the consumer brand corn that's been around since the 1970s, that's an example of one mycoprotein. Very interesting because it has a kind of fibrous texture and is accepted by consumers. But we think that this, the fact that there's one example out there is wonderful, but it's still a largely untapped area. We haven't looked into other bacterial yeasts or fungal protein sources with any sort of focus in the last years. And so that work is kind of ongoing to screen for different strains. So I'm going to switch gears now and go a little bit into the plant-based foods and talk you through some of the developments with regard to plant-based foods. I mean, the first thing to say is there's a tendency to talk about, is a protein functional? And I think it's important to say every protein can be functional. We just need to do a proper matching of the specific protein to the need that we have. And a protein that's functional in a beverage, of course, has to be sort of highly soluble and not sink to the bottom. But if we look into, for example, a meat analog, or if we're trying to make a plant-based sausage, we need a very different performance from that protein. So it's really a kind of matchmaking process. If we start from a particular source, we can look for, okay, what might we reasonably expect from this protein at the end of the pipe? Or vice versa, if we start from a specific application area, then we sort of work backwards, asking ourselves what kind of proteins can deliver this functional performance. If we look into first dairy alternatives, one of the sort of big issues that we're working to tackle with dairy alternatives is making them sort of cleaner label and better nutrition, more nutritious by enabling actual protein gels to be prepared. So if you pick up a plant-based dairy, a plant-based yogurt nowadays, you typically will see a long list of starches and polysaccharides, and we're actually not we're actually not producing a protein gel in those plant-based yogurts. So they're not very analogous to a dairy yogurt where that sort of gel is completely protein dependent, made from casein from dairy proteins. There's a reason for that. The currently available plant proteins tend to be less soluble and less able to gel, so significantly less functional in this sort of acid set gel than dairy proteins. Part of that is due to the primary processing, the way we're extracting those proteins. So we do believe that we need to sort of go back to how those proteins are extracted if we want to be able to get them to perform in a dairy alternative. Our work in enabling this rests on 3 pillars. One, sort of milder primary processing to maintain the inherent capability of the protein. Number 2, sort of understanding synergies between plant protein sources. We often see that we need 2 plant proteins to get a job done. Most of the meat analogs and dairy analogs out there, you'll see a combination of sources. And number 3, understanding the interactions between the plant proteins and other components. There's often a higher level of fiber in those plant materials than there is in dairy proteins, for example. We need to understand how to cope with that. So I'll just show you in 2 slides some work that we're doing in these directions. So number 1, when we look at milder processing, we've developed a sort of proof of concept of how we can extract proteins from mung beans using industrially relevant and standard unit operations to create microgels, which can be concentrated and dried into an ingredient that enables a heat set gel preparation, which is a true plant-based protein gel. And then the other very important direction, as I mentioned, was looking at the sort of synergies between different proteins. And what I want to show you in this slide is, depending on how we're pre-treating proteins together, we can create very different microstructures. So if you sort of look at these colorful pictures, these are confocal microscope images of gels. And you can see that, in some cases we've got microphase separation happening, and in others we see a relatively sort of homogeneous gel structure. And if you look at this chart on the right side, you can see that corresponds to very different firmness levels of the gels. And I want to be clear, we used different pHs, but that's a pre-treatment pH. So all of these are prepared into a yogurt-like gel. So it's really only about when we heat treat it, minor differences in pH are creating major differences in the microstructure and ultimately in the gel texture of these combinations of proteins. And we have a project that kicked off earlier this year that's looking, a 4-year project that's looking to develop functionality maps of combinations of different plant proteins and sort of understanding when do they interact homogeneously, when do they have microphase separation, and how does that impact their texture. Then if we look at the other important application domain, if we look briefly at meat analogs, we believe that these products are very helpful in creating a driver to the transition for the consumers who traditionally enjoy consuming meat. Most or almost everything on the market today is produced using one of 3 routes. Number 1, mixing and forming. If you've got something like a sausage, it's typically done, made in this direction. Low moisture extrusion, where we put a dough through an extruder with a relative low moisture content. Sorry, that's kind of obvious. And then we get a kind of a textured vegetable protein out of that, which is then mixed and formed into a burger patty. If you've bought a plant-based burger in the market, it's probably made using this technology. And then last but not least, high moisture extrusion, where we put a dough through an extruder with a slightly higher level of moisture, and we get a typical product that looks a bit like the picture there, something that looks more like a chicken tender. Within our organization, we have work on looking at improved technology directions, and in particular, we challenge ourselves to see if we can create a plant-based steak. We believe that high moisture extrusion is getting us pretty close on chicken, but a whole cut of meat like a steak is still a little bit beyond what we can achieve with an extruder. So we've invented a new device called a shear cell, which provides a higher degree of control during processing, and thereby also a higher degree of fiber alignment And you can see in the picture that we get a very steak-like texture out of this process. We're very satisfied with the texture and bite that comes out of this process. We're still working on juiciness. I think that's still a big challenge for all of the plant-based products. And we'd also like to, in particular, get toward the ability to apply less refined ingredients. We still see that a lot of the texture development in meat analogs is dependent on having quite purified protein ingredients. So we have now in development a next generation of public-private partnership projects, and I just wanted to end by sharing with you some of the next goals that we have in mind. We have a project called Analogs for All, where we're looking for low energy alternatives to extrusion. We see that extrusion capacity globally is a limiting factor for meat analogs. We have a project called Intrinsic, where we are looking at biorefinery of oil and protein ingredients from oil seeds, for example, rapeseed. We're very interested in how we can valorize the proteins from that, which is a big volume industrial residue right now. We have a project called Informed, which is sort of bundling together the needs to optimize plant-based products through fermentation, focus mainly on flavor, but also on texture and shelf life, how we can improve those qualities using fermentation. And last but not least, very importantly, a project Health and Analogs, where we're evaluating and looking to improve the health aspects of meat analogs. While we think that they're an important part of the transition, many of them are not optimal in terms of their nutritional quality. And actually, there's a lot we don't know about the health biomarkers of the consumers who are buying these products right now. What's its impact on our cardiovascular health? What's its impact on the gut microbiome, for example? And my very last slide, this is what we're working toward in 2050. I think the oceanic farming was very well covered by my predecessor, so wonderful for that. We do expect to see a lot more pulse and grain production on the land. We're working toward a rebalancing in the consumption of animal sources and plant-based, so that we get back to something that we were closer to 60 years ago. And we are very committed to working toward a fully circular food system where we have, for example, upcycled those agricultural and industrial residues into food where possible and feed when not. So with that, I'd like to thank you very much for your attention, and I'm happy to take questions.